In the next few minutes, you're going to have a thorough understanding of what monoclonal antibodies are and how they're produced in a laboratory. This is quite topical because of the ongoing COVID-19 pandemic. In order to understand what monoclonal antibodies are, we'll have to first look at a few basic concepts. So let's begin. Antibodies act like superheroes that protect our body from the bad guys, in this case, the antigens. Let's take a closer look at how an antibody recognizes an antigen. Antibodies do not recognize an antigen as a whole. Rather, they recognize certain specific portions or sites on the antigen, thereby stimulating an immune response. These specific portions or sites are called epitopes or antigenic determinants. Epitopes are protein units that consist of approximately 5 to 8 amino acids. Epitopes are generally divided into two categories. Continuous epitopes, where amino acids that are to be recognized by the antibody are present one after the other or in a sequential manner. Or conformational epitopes, where amino acids that are to be recognized by the antibody are not present in a sequential manner but are brought together by protein folding. A point to keep in mind is that antibodies are produced by a very important component of our immune system called the B cells. Let's now look at what monoclonal antibodies are. The simplest definition can be derived from the word monoclonal itself, meaning single clone. So if you look at the figure here, this is a B cell. This B cell is cloned to produce several exactly similar B cells. The antibodies generated by these structurally and genetically exactly similar B cells are called monoclonal antibodies. Since their parents are identical, they would be identical as well. Another point to keep in mind is that monoclonal antibodies of a particular type would recognize the same type of epitope. This is a depiction. Here you can see that the red antibodies are products of a single type of B cell and hence recognize only the red colored epitope shown here. Whereas the blue colored antibody is produced from another type of B cell clone and hence can recognize the blue colored epitope that is shown here. So how exactly are these monoclonal antibodies made in a laboratory? The first step is to immunize an organism with a specific antigen. Most commonly, mice are used in such experiments because of their physiological similarity with humans. The injection is done intraperitoneally, that means that it is injected onto the abdomen of the mouse. Usually, several rounds of immunization are carried out over a period of time to achieve the required antibody titer. Once the mouse's system recognizes the antigen injected into it as foreign, the B cells will start producing antibodies. Now, if you look at the antigen, you can see that there are several epitopes present on it, and hence, the B cells would produce antibodies specific to each of these epitopes. After this step, blood samples are taken from the mouse to check if the target antibody is being produced. Techniques like ELISA are used to determine this. In the next step, the B cells are isolated from the spleen of the mouse and moved into a culture. Now, there is a problem that arises at this step. B cells cannot be cultured for long as they have a very short lifespan. Since our aim here is to proliferate or clone these B cells over and over again in order to produce a large volume of monoclonal antibodies, this short lifespan problem has to be solved. One way we can do this is by fusing these B cells with another type of cell line called myeloid cells, which are immortal cells. So this fusion will give rise to cells that are both antibody producing as well as immortal. Polyethylene glycol is one of the chemicals that can be used to obtain this fusion. After this step, we can now expect around 5 different cell types in the culture media. There can be B cells that did not fuse with any other cell and remained unfused. There could be myeloid cells that did not fuse with any other cells and remained in its original unfused form. There could be B cells that fused with each other in a homogeneous manner. There could be myeloid cells that fused with each other in a homogeneous manner. And then there could also be a hybridoma, which is our target cell, which formed by the fusion of a B cell and a myeloma cell. Now, we don't want any of these cells competing with our target hybridoma cells for nutrients. Hence, we need to remove all the cells from the culture media except for our target hybridoma cell. This can be done by using a selection process that requires a medium called HAT medium. HAT stands for hypoxanthin aminopterin thymidine. In order to understand this selection process, we need to keep in mind that when a cell proliferates or divides, it replicates its DNA. This is done either by using the de novo pathway or by using the salvage pathway. De novo means from scratch, and as the term suggests, this particular pathway produces nucleotides required for replication from scratch. The aminopterin present in the HAT medium blocks this particular de novo pathway. This means that all the cell types in the medium would now have to undergo the salvage pathway to produce their nucleotides required for replication. The salvage pathway requires three important components. It requires hypoxanthin and thymidine, which is present in the media, and it also requires an enzyme called HGPRT. Now, our myeloid cells do not have the enzyme HGPRT and hence cannot employ the salvage pathway, and hence these will die out. And now the B cells that are present in the medium, as I mentioned earlier, they have a very short lifespan and these die out from the medium as well. So now the HAT medium has selected the hybridoma cell alone, which has the HGPRT enzyme and also has hypoxanthin and thymidine obtained from the medium. These hybridoma cells proliferate rapidly and produce a large volume of highly specific monoclonal antibodies. These monoclonal antibodies have a lot of application in both diagnostics as well as in the medical field. A lot of research is now being targeted at producing monoclonal antibodies that would help curb the effects of COVID-19. The coronavirus gains entry into the human cell with the help of one of its structural proteins, which is called the spike protein, that binds to one of the receptors present on the human cell called ACE2. It does so with the help of a receptor binding domain, S1, which attaches to the ACE2 receptor on the host cell in a key and lock fashion. This results in fusion between the virus and the host cell, and thereby it allows the entry of the virus into the human cell. Monoclonal antibodies that could specifically target the receptor binding domain on the spike protein of the coronavirus would block it from being able to bind to the ACE2 receptor on the human cell. This would prevent it from entering the cell and causing disease. Thank you so much for watching. Please hit like and subscribe.